Since the dawn of humanity, we have gazed at wounds and wondered, how can we heal? In the flicker of firelight, our ancestors painted stories of pain and hope on cave walls, searching for answers in nature and spirit. Across continents, healers gathered roots and herbs, whispered prayers, and performed rituals, each culture weaving its own tapestry of medicine. From the wisdom of ancient Egypt and China to the scrolls of Greece and Rome, knowledge was passed from hand to hand, generation to generation. Over centuries, curiosity and resilience sparked revolutions. Alchemists became scientists, mystics became physicians. The quest to heal drove us from herbal brews to antibiotics, from sacred rituals to gene therapy. Each breakthrough a testament to our ingenuity and compassion. Today we stand on the shoulders of those who came before, explorers of the unknown, healers of the body and mind. Join us as we journey through the evolution of medicine, discovering how the story of healing is, at its heart, the story of us all. Egyptians and Mesopotamians, herbs, surgery, spiritual rituals, Ayurveda and traditional Chinese medicine, TCM, systematic approaches to diet, lifestyle herbs, acupuncture, Greek and Roman medicine, Hippocrates' humors, Galen's anatomy, indigenous knowledge, plant-based cures, holistic health. Ancient medicine combined observation, trial and error, and spiritual belief. While limited by science, many traditions laid the foundation for modern pharmacology. As the Roman Empire crumbled, much of its vast medical knowledge fractured and faded in the West. Europe entered the Middle Ages, a period where medicine became deeply entwined with faith. The prevailing belief, propagated by the Church, was that illness was not merely a biological malfunction but a spiritual one, a punishment for sin, a test from God, or the work of demonic forces. In the quiet cloisters of monasteries, ancient knowledge was preserved, but selectively. Monks became the primary custodians of healing, tending to the sick in infirmaries. Their practice was a blend of herbal remedies, prayer, and a simplified dogmatic version of Galenic theory. The theory of the four humors, blood, phlegm, yellow bile, and black bile, remained the unchallenged foundation of physiology. Health was a state of perfect balance, sickness, and imbalance. Treatments aimed to restore this equilibrium through methods like bloodletting, purging, and specific diets, all administered alongside fervent prayer and appeals to saints. But this fragile system of faith and humoral theory was about to face a cataclysm that would bring it to its knees. In 1347, Genoese trading ships docked in Sicily carrying not just cargo but a terrifying unseen passenger. Yersinia pestis, the Black Death, had arrived. The bubonic plague swept across the continent with terrifying speed and lethality. It was a force of nature that defied prayer and medical intervention. Symptoms were horrific: fever, vomiting, and the telltale buboes, swollen blackened lymph nodes in the groin and armpits that gave the plague its name. Within a few years, it had wiped out an estimated 1/3 to 1/2 of Europe's population. Society teetered on the brink of collapse. The sheer scale of death was incomprehensible, shattering families, economies, and the very authority of the Church and its medical doctrines. The failure of medicine during the Black Death was absolute. Physicians relying on theories of miasma or bad air could do nothing. The Church, which taught that suffering was divine will, could offer no tangible relief. This created a profound crisis of faith and authority. People began to question the established order, and this questioning, born from immense tragedy, inadvertently planted the seeds for a new way of thinking about the human body and disease. While Europe grappled with plague and dogma, a different story was unfolding across the Mediterranean. In the Islamic world, a vibrant intellectual renaissance was in full swing. From the 8th to the 13th centuries, scholars in cities like Baghdad, Cairo, and Cordoba embarked on a monumental effort to gather, translate, and build upon the knowledge of the ancient world. They didn't just preserve the works of Galen and Hippocrates, they critically analyzed, tested, and expanded upon them. Figures like Al-Razi, or Rhazes in the 9th century, wrote extensively on clinical observation, famously being the first to systematically differentiate between smallpox and measles. But it was an 11th century Persian polymath who would create one of the most influential medical texts in history. His name was Ibn Sina, known to the West as Avicenna. His masterwork, the Canon of Medicine, was a colossal 5-volume encyclopedia. It was a systematic compilation of all contemporary medical knowledge, from detailed anatomy and pharmacology to complex diagnoses and surgical procedures. Avicenna discussed the contagious nature of diseases like tuberculosis, the use of anesthesia, and even proposed methods for clinical drug trials. For over 600 years, the canon would serve as the definitive medical textbook, not only in the Islamic world but also in European universities, once it was translated into Latin. This intellectual flourishing was matched by institutional innovation. The Islamic hospital, or Bimaristan, was a marvel of its time. The Al-Mansuri Hospital in 13th century Cairo, for example, was a vast, organized complex with separate wards for different illnesses, running water, a pharmacy, a library, and an attached medical school. It was a secular institution, dedicated to healing all patients regardless of their background, and served as a center for treatment, education, and research. A model that starkly contrasted with the more rudimentary hospices of Europe. The Bimaristan represents a fundamental shift. It institutionalized the idea that medicine should be based on rational care and continuous learning. The combination of a hospital, a medical school, and a library in one place created a powerful ecosystem for advancing medicine. The knowledge and practices developed here would eventually flow back into Europe, providing a critical foundation for the changes to come. As the Middle Ages waned, Europe began to awaken. The rediscovery of classical texts, many preserved and transmitted through the Islamic world, fueled a new philosophy, humanism. This was a celebration of human potential, intellect, and crucially, direct observation of the natural world. And nothing was more central to this new focus than the human body itself. For centuries, human dissection had been a taboo, shrouded in religious and cultural restrictions. Early university dissections were often sterile performances, where a professor would read from an ancient Galenic text while a barber surgeon made the cuts, with no one daring to point out discrepancies between the text and the body on the table. But in the 16th century, a young, ambitious anatomist from Brussels would shatter that tradition. His name was Andreas Vesalius. As a professor in Padua, one of Europe's leading medical centers, he did something revolutionary. He stepped down from the professor's chair, took the knife in his own hands, and trusted the evidence of his own eyes over the dogma of a 1300-year-old text. What he found was astonishing. Galen, who had based his anatomy on the dissection of animals like apes and pigs, had made hundreds of errors when applying it to humans. Vesalius discovered the human jaw was a single bone, not two. The sternum had 3 parts, not 7. The liver had 2 lobes, not 5. In 1543 he published his findings in a monumental work, De Humani Corporis Fabrica, on the fabric of the human body. Aided by the printing press, the book's breathtakingly detailed and artistic illustrations brought human anatomy to life with unprecedented accuracy. It was a declaration of war on ancient authority and a founding document of modern observational science. It's hard to overstate the courage of Vesalius. He wasn't just correcting a few facts, he was challenging the entire philosophical basis of medicine. He was saying that truth comes from empirical investigation, not from received wisdom. Every medical student today who steps into an anatomy lab is walking in Vesalius's footsteps. The Fabrica established anatomy as the bedrock of medical education, the essential map without which no physician can navigate. This period, stretching from the medieval monastery to the Renaissance anatomy theater, was an era of profound and often violent contrast. Superstition coexisted with science, faith-based healing was practiced alongside the first clinical trials. While alchemists sought the elixir of life, surgeons like Ambroise Paré were pioneering new techniques on the battlefield, abandoning brutal cauterization for the gentle art of ligating arteries. The Black Death, for all its horror, exposed the limits of dogma. The Islamic Golden Age served as the world's intellectual custodian, preserving and advancing knowledge when it was lost to the West. And the Renaissance, championed by figures like Vesalius, finally reintroduced the most powerful tool in medicine: direct, unbiased observation. This was not a simple linear progression, but a complex interwoven story of faith, plague, and the revolutionary act of looking for oneself. It was the difficult, bloody, and absolutely essential birth of the modern medicine we know today. For millennia, the practice of medicine was shrouded in a fog of tradition and theory, largely disconnected from the biological reality of the human body. The prevailing doctrine, inherited from the ancient Greeks, was humoral theory. It posited that health depended on the delicate balance of 4 bodily fluids or humors: blood, phlegm, yellow bile, and black bile. Illness was seen as a disruption of this balance, an internal disharmony. Treatments, therefore, were aimed at restoring equilibrium through methods that seem crude to us today. bloodletting, purging, and dietary restrictions, all designed to remove a supposed excess of one humor or another. Disease was not an external invader, but an internal imbalance. The cause of great plagues and fevers was often attributed to miasma, a noxious form of bad air emanating from rotting organic matter. While these theories were born from careful observation of symptoms, they lacked a fundamental understanding of the true cause of illness. A revolution was needed not of politics or nations but of perception itself. It would begin not in a grand university or a royal court, but in the hands of a humble cloth merchant in the Dutch city of Delft. Antony van Leeuwenhoek was not a trained scientist. He was a draper, whose obsession with quality control led him to develop his own powerful single-lens microscopes to inspect the threads of his fabrics. But his boundless curiosity soon turned his lenses from textiles to the world around him. In the 1670s he began examining everything he could find: rainwater, dental plaque, blood, and infusions of pepper. What he discovered was a world teeming with life, previously invisible and entirely unimagined. He called these creatures animalcules, or little animals. In letters to the Royal Society of London, he described them with meticulous wonder. "I saw with great wonder very many very little living animalcules, very prettily a-moving." He had opened a window into the microbial universe. Yet, for all his groundbreaking observations, the crucial connection was not made. Leeuwenhoek saw these microbes, but he did not, and could not, comprehend them as the agents of disease. The concept was simply too alien. His discovery remained a curiosity, a marvel of the natural world, but its medical implications lay dormant for over a century and a half, waiting for new minds to bridge the gap between seeing and understanding. The first major, albeit tragic challenge to the old ways came not from a laboratory but from a maternity ward. In the 1840s, the Vienna General Hospital was a place of both hope and terror. It housed 2 maternity clinics. In the first clinic, staffed by physicians and medical students, mortality from puerperal fever or childbed fever was horrifically high, sometimes reaching 30%. In the second clinic, staffed exclusively by midwives, the rate was a fraction of that. A young Hungarian doctor named Ignaz Semmelweis was tormented by this discrepancy. He systematically eliminated potential causes from birthing position to atmospheric influences. The terrible truth was revealed when a colleague died from an infection after being accidentally cut by a student's scalpel during an autopsy. The colleague's own autopsy revealed pathology nearly identical to that of the women dying from childbed fever. Semmelweis had his epiphany. He and his students were carrying cadaverous particles on their unwashed hands from the autopsy room directly to the mothers in the delivery ward. He instituted a strict policy. All physicians must wash their hands in a solution of chlorinated lime before examining patients. The results were immediate and staggering. The mortality rate in his clinic plummeted to below 1%. But Semmelweis had not discovered a germ. He had only implicated an unseen something. His evidence was statistical, not biological. The medical establishment, insulted by the implication that they, the healers, were the cause of death, viciously rejected his findings. Semmelweis was driven from Vienna, his work ignored, and he died in an asylum, ironically from the very type of infection he had fought so hard to prevent. His story is a tragic lesson in how entrenched dogma can resist even the most obvious evidence. The definitive proof would come from France. Louis Pasteur, a chemist, was initially tasked with solving a practical problem for the French beverage industry. Why were wine and beer sometimes spoiling? Through his microscope, he discovered that fermentation was caused by living microorganisms, yeasts. Spoilage, he found, was caused by other, undesirable microbes. He developed a process of gentle heating to kill these organisms, a process we still call pasteurization. This work led him to a profound conclusion. If microbes could cause changes in wine, could they not also cause changes, that is, disease, in the human body? He set out to disprove the long-held theory of spontaneous generation, the idea that life could arise from non-living matter. His elegant swan neck flask experiment was definitive. Broth boiled in a flask with a long, curved neck remained sterile indefinitely because microbes from the air were trapped in the bends. But if the neck was broken, allowing dust and air direct access, the broth quickly teemed with life. Life, he proved, only comes from life. This was the theoretical death blow to miasma and the birth of the germ theory of disease. While Pasteur laid the theoretical groundwork, it was a German country doctor, Robert Koch, who provided the rigorous, systematic proof. Koch was a master of laboratory technique. he pioneered methods that are still fundamental to microbiology today. Frustrated with trying to study bacteria in liquid broths where different species were jumbled together, he developed a way to grow them in pure colonies on a solid medium, first using a potato slice, and later, on the suggestion of his assistant's wife, Angelina Hesse, using agar. This allowed him to isolate a single type of bacterium and study it in detail. With these methods, he developed a set of criteria, a scientific gold standard known as Koch's postulates, to prove that a specific microbe causes a specific disease. First, the microorganism must be found in abundance in all organisms suffering from the disease, but not in healthy organisms. Second, it must be isolated and grown in a pure culture. Third, the cultured microorganism should cause the same disease when introduced into a healthy organism. And fourth, the microorganism must be re-isolated from the inoculated, diseased host, and identified as being identical to the original specific causative agent. Using this powerful logic, Koch systematically unmasked the invisible killers of his time. In 1876, he identified as the cause of anthrax. In 1882, he stunned the world by identifying the agent of the White Plague, tuberculosis. A year later, he identified during an epidemic in Egypt. The age of mystery was over. The enemy now had a face. As the 19th century progressed, this new scientific understanding began to yield powerful new tools. The first of these, however, predated the full acceptance of germ theory, arising instead from keen observation and daring experimentation. Smallpox was one of history's greatest scourges, a viral disease that killed nearly a third of its victims and left survivors permanently scarred. An English country doctor named Edward Jenner noticed a piece of local folklore. Milkmaids who contracted cowpox, a mild disease from cattle, seemed to be immune to smallpox. Jenner hypothesized that the cowpox infection provided protection. In 1796, in a procedure that would be ethically unthinkable today, he decided to test his theory. He took pus from a cowpox sore on the hand of a milkmaid and inserted it into scratches on the arm of an 8-year-old boy, James Phipps. The boy developed a mild fever but quickly recovered. Several weeks later, Jenner exposed the boy repeatedly to smallpox. James Phipps never became ill. Jenner had performed the world's first vaccination, a term he coined from the Latin word for cow. This single act laid the foundation for immunology and demonstrated that the body could be trained to fight off an invader, heralding a future where diseases could be prevented, not just treated. While prevention was a monumental step, the realm of surgery remained a house of horrors. Before the mid-19th century, surgery was a brutal, terrifying last resort. Operations were performed on fully conscious patients whose screams filled the theater. The best surgeon was the fastest, a human sawbones who could amputate a leg in under 3 minutes to minimize the agony. The primary barriers were pain and shock. This changed on October 16, 1846. In the operating theater of Massachusetts General Hospital, a dentist named William T.G. Morton administered sulfuric ether to a patient, who then underwent the removal of a neck tumor without pain or struggle. The age of anesthesia had begun. Chloroform soon followed. Surgeons, now freed from the tyranny of the clock, could perform longer, more intricate and more ambitious procedures. But a dark paradox emerged. As operations became more complex, post-operative mortality rates soared. Patients who survived the knife would die days later from rampant infections. This was the black period of surgery. The solution came from Joseph Lister, a surgeon in Glasgow who had read Pasteur's work. He theorized that the same microbes causing fermentation were causing wounds to putrefy. He began experimenting with carbolic acid as an antiseptic, applying it to dressings, spraying it in the air, and using it to sterilize his instruments. The results were miraculous. His wards, once filled with the stench of gangrene, became places of recovery. Lister's principle of antisepsis, which would evolve into the modern practice of aseptic surgery, finally made the interior of the human body a safe domain for healing. The final piece of the puzzle was not about treating individuals but protecting entire populations. The Industrial Revolution had created cities that were breeding grounds for disease. Overcrowded, with no concept of sanitation, their water sources were often contaminated by human waste. Cholera and typhoid epidemics swept through these cities, killing thousands. During London's devastating cholera outbreak of 1854, a physician named John Snow conducted a brilliant epidemiological investigation. Rejecting the dominant miasma theory, he suspected the water. He painstakingly mapped the locations of the deaths and found they were all centered around a single public water pump on Broad Street. His investigation revealed the pump was contaminated by a nearby cesspool. By convincing the local authorities to take the radical step of removing the pump handle, Snow stopped the outbreak in its tracks. He didn't need to see the cholera bacterium. His methodical mapping of the disease's pattern was enough. His work founded the science of epidemiology. This, combined with events like London's Great Stink of 1858, when the stench from the Thames became so unbearable it shut down Parliament, finally spurred massive public works projects. The creation of comprehensive sewer systems like the one engineered by Joseph Bazalgette in London, and the establishment of clean water supplies were perhaps the single greatest medical advance of the era, saving more lives than any single doctor. The 18th and 19th centuries thus witnessed the complete transformation of medicine. It was a paradigm shift of epic proportions, moving the art of healing from a realm of ancient tradition and speculative philosophy into the domain of rigorous, evidence-based science. The discovery of the microbial world provided a tangible enemy. The development of vaccination, anesthesia, and antiseptic surgery provided powerful new weapons, and the rise of public health and epidemiology provided the strategy to protect entire societies. This period laid the very foundation upon which all of modern medicine is built. It established a new covenant, that through observation, experimentation, and the relentless pursuit of evidence, illness was no longer an unknowable mystery or a divine punishment, but a biological problem that humanity could and would solve. Antibiotics. Alexander Fleming discovers penicillin, 1928. Vaccines expand. Polio, measles, hepatitis, medical technology, X-rays, MRI, Connecticut scans, dialysis, pharmaceutical industry, mass production of drugs but also issues of overuse and side effects. Ethics and reforms, Nuremberg Code, bioethics, informed consent. The 20th century gave us miracle drugs and technology, but also taught us lessons in ethics and responsibility. As the 21st century dawned, medicine stood on the precipice of a monumental leap, one that would shift its very foundations. The catalyst was the Human Genome Project. Concluded in 2003 after 13 years of intensive international collaboration, this audacious endeavor mapped the entirety of our genetic blueprint, all 3 billion base pairs. It was the biological equivalent of the Apollo program, a moonshot that gave us the complete instruction manual for building a human being. Initially, this vast sea of data was overwhelming. But as technology advanced, the HGP's true legacy emerged. It wasn't just a map, it was the bedrock for an entirely new philosophy of care: personalized medicine. The cost of sequencing a genome plummeted from billions of dollars to mere hundreds, transforming a massive research project into a viable clinical tool. For the first time, we could move beyond treating symptoms and begin to understand and even predict disease at its most fundamental level, based on an individual's unique DNA. This wasn't just an evolution, it was a revolution in how we perceive human health and illness. If the Human Genome Project gave us the map of life, the discovery of CRISPR-Cas9 handed us the pen. Derived from a defense mechanism in bacteria, this revolutionary gene editing tool acts like a biological word processor with a find and replace function for DNA. The system uses a guide RNA to locate a specific faulty gene, and the Cas9 enzyme then acts as molecular scissors, cutting the DNA at that precise spot. This allows scientists to delete, repair, or replace the defective gene. The therapeutic potential is staggering. For monogenic diseases, illnesses caused by a single genetic mutation, CRISPR offers the tantalizing possibility of a true cure. Clinical trials are already underway for conditions like sickle cell anemia and beta thalassemia, where editing a patient's own stem cells could permanently correct the disease. However, this immense power brings with it profound ethical questions. The 2018 announcement of the world's first gene-edited babies in China ignited a global firestorm. It crossed the critical line from somatic editing, which affects only the patient, to germline editing, which alters the human gene pool for all future generations. Where do we draw the line between therapy and enhancement? Who decides what genetic flaws should be corrected? These are no longer theoretical questions, they are urgent challenges that society and the medical community must navigate with extreme caution. While genomics looked inward at our code, another revolution was brewing. One that weaponized our own bodies against our most feared disease, cancer. For a century, the pillars of oncology were surgery, radiation, and chemotherapy, brute force methods that cause significant collateral damage. Immunotherapy represents a paradigm shift. Instead of attacking cancer directly, it empowers the patient's own immune system to do the job. We discovered that many cancers survive by exploiting natural checkpoints on immune cells, effectively hitting the brakes on T cells that would otherwise destroy them. Checkpoint inhibitor drugs release these brakes, unleashing the immune system to hunt down and eliminate tumors. The results can be dramatic, leading to long-term remissions in previously untreatable cancers like metastatic melanoma. An even more targeted approach is CAR T-cell therapy. Here, a patient's T cells are extracted, genetically engineered in a lab to produce chimeric antigen receptors or CARs that are specifically designed to recognize their cancer and then reinfused. The story of Emily Whitehead, the first child to receive CAR T therapy for leukemia, is a testament to its power. After relapsing from all conventional treatments, she was given this experimental therapy and made a complete recovery. It's a complex, expensive, and sometimes dangerous treatment, but it has proven that a living, targeted drug can succeed where all else has failed, revolutionizing hematologic oncology. The explosion of data from genomics, proteomics, and electronic health records created a new challenge: information overload. This is where artificial intelligence and big data enter the medical arena. Machine learning algorithms can sift through datasets of a scale no human could ever comprehend, identifying subtle patterns that are invisible to us. In diagnostics, AI is transforming radiology and pathology. Trained on millions of images, these systems can detect diabetic retinopathy, skin cancers, or early signs of tumors on a CT scan with a level of accuracy that often meets or exceeds that of human experts, acting as a crucial second opinion. Beyond diagnostics, AI is becoming predictive. By analyzing a patient's health records, genetic predispositions, and lifestyle data, it can forecast the likelihood of developing conditions like sepsis or heart failure, enabling clinicians to intervene proactively. In the operating room, robotic surgery has enhanced the surgeon's capabilities, offering greater precision, stability, and dexterity. Systems like the Da Vinci robot translate a surgeon's hand movements into micro-movements, filtering out tremors and allowing for minimally invasive procedures that reduce pain, blood loss, and recovery time. AI is not replacing doctors, it is augmenting them, providing powerful new tools for diagnosis, prediction, and treatment. Yet for all our technological prowess, the 21st century has also magnified our vulnerabilities. We face global health challenges that threaten to undermine our progress. Chief among them is antibiotic resistance. Decades of overuse and misuse in both medicine and agriculture have accelerated natural selection, breeding superbugs resistant to our entire arsenal of antibiotics. We risk sliding back into a pre-antibiotic era where routine surgeries become life-threatening and a simple cut can lead to a fatal infection. The threat of pandemics has also become a stark reality. In an interconnected world where a virus can cross continents in hours, outbreaks of SARS, MERS, and most devastatingly, COVID-19, have exposed the fragility of our global health systems. While the rapid development of mRNA vaccines was a scientific triumph, the pandemic laid bare our failures in coordination, preparedness, and equity. This brings us to the most pervasive challenge of all: inequality. The miraculous breakthroughs, gene therapies costing millions, advanced robotic surgeries, personalized cancer treatments, are largely accessible only to the wealthiest individuals in the wealthiest nations. This growing chasm between what is medically possible and what is globally available creates a deep ethical crisis. How can we claim to be in a golden age of medicine when life-saving innovations remain out of reach for the vast majority of the world's population? We are undeniably in a new era of medicine. The one-size-fits-all model is giving way to a new paradigm, often called P4 medicine. It is predictive, using genomics and AI to forecast illness. It is preventive, enabling early intervention. It is personalized, tailoring treatment to the individual. And it is participatory, empowering patients to be active partners in their own health. The power at our disposal is unprecedented. We can edit the code of life, reprogram our own immune cells, and operate with superhuman precision. But this bright future is shadowed by the immense challenges of ethics, access, and equity. The defining task for the next generation of physicians, scientists, and policymakers will not just be to invent the next miracle cure, but to build a system that can deliver these miracles to all who need them. The story of medicine is a story of relentless progress, but its next chapter must be a story of shared progress. The future of health depends not only on the brilliance of our science, but on the breadth of our compassion. From herbs to AI, medicine has evolved with us. The journey is far from over. The next frontier, longevity, prevention, and precision medicine, will shape not just how long we live, but how well we live. The evolution of medicine is ultimately the story of humanity's survival.